And so in today's webinar, we will be tackling the following topics. We will be providing you with an overview of the IVDR. We will also get into the details of the Article 5.5. We will get you through the topic of method development and the approach, whereby then how do we link this with the regulatory strategy, touch on the design control phase, as well as the method validation, followed by the technical dossier. And all this, we want to wrap it up by sharing with you our lessons learned, and then obviously having a panel discussion on questions that will be raised during the webinar. And so therefore, as we all know very well today, IVDR is a European regulation, and it is applied specifically to Europe as well as the European Economic Area. However, IVDR reach extends well beyond Europe. Any manufacturer or organization wanting to plan IVDs on the European market or conduct a clinical study within the EU sites, they must comply. And therefore, as a result, IVDR has effectively become a global benchmark for diagnostic regulations. So the IVDR is a European regulatory framework that governs all in vitro diagnostic devices, whether they are used in clinical trials or whether they are used in routine patient care. Its objective is to ensure a higher level of safety, performance, and transparency, and that is across the entire life cycle of the diagnostic tests. It introduces more stringent requirements, particularly around the clinical evidence, as well as the quality systems and the oversight that is being put in place. And therefore, as a result, both the manufacturers as well as the laboratories, they are seeing, well, all of us together, we are seeing a significant shift in how diagnostic assays are developed, validated, and being used. With this context in mind, I want to start this webinar by providing you a very high-level practical overview, which highlights how do we select and justify the use of an IVD in the context of a clinical trial. And so therefore, we've put together a flowchart which captures a key decision that laboratories and study teams will have to early on rely on. The first thing is, do we use an existing CE-IVD marked test, or do we proceed with a laboratory developed test under Article 5.5? If a suitable CE-IVD kit is already available, then we take the path of Route 1 that is presented on this flowchart, which now it's really a relatively straightforward path. The laboratory can use the assay as intended by the manufacturer without any modification, relying on the fact that the vendor has already established the IVDR compliance. And now this significantly reduces the regulatory burden on the lab. However, if there is no appropriate CE-IVD option that exists on the market, then we are now embarking into Route 2, which is presented on this flowchart. And this means now that the laboratories must take a much greater responsibility. And this requires now meeting specific conditions under the LDT pathway. And these special conditions include implementation of the ISO 15189, quality compliant with a quality management system, ensuring the general safety and performance requirement, the GSPR compliance, as well as issuing a public declaration documenting adherence to the regulatory requirements. Therefore, for today's webinar, we will focus on Route 2, the custom LDT approach, which is quite common today in our laboratories because we oftentimes have new type of assays and as well as into emerging areas. We will take you through the regulatory component And we will also use an example of an immunoassay that will then expose to you what is required from a technical performance. And so I will now hand off the talk to Marie-Laure Delalande, who will start off with an overview of the process.
I will present now the IVDR context under Article 5.5. The slides gives you the map for today's case study. We have 3 phases here. Phase 1 is about method developments. Start by optimizing and reproducing the assay, then identifying the critical step that could cause variability. Phase 2 is the strategic pivot. There is where we define the regulatory strategy, in our case, the Article 5.5 route under IVDR. Phase 3 is where the real work happens. Design control first, method validation, and finally the technical dossier. 3 milestones mark the progress: method development complete, regulatory strategy set, and the full technical package ready. Everything we discussed today sits within this framework. Let's start with the regulatory strategy. Before we run a single experiment, we need to answer 3 questions. First, the route. Here, under Article 5.5 exemption, as Lorella mentioned previously. Second one, what are the GSPR requirements? The GSPR requirements in Annex 1 of the IVDR. Every LDT must address this GSPR. They define what good enough looks like from a regulatory standpoint. Third, is Article 5.5 actually applicable? We need to formally confirm eligibility. The test stay in-house. The lab is ISO 15189 accredited, and we can justify that no equivalent study exists in the market. Then we move to the scientific validity. This means documenting with literature and clinical data that the assay actually measures what it claims to measure. For this assay, that means linking the measure of the titer to clinical outcomes in the therapy patients. Finally, the design control loop. This is the structural framework that connects everything from patient need to validate assay. We will come back to this in detail. Before, let me pass the floor to Martin for the next part.
Thank you, Marie-Laure. So in the next slide, we're going to start by talking about an example of the assay that was going to be used today to present a little bit of a case study. So this is really a generic example of a typical ligand binding assay that uses a custom format. So in this assay, you can see that there's a coded reagent, which is used as the capture reagent, and there is also a detection reagent for detection of the signal. And the reagent is first coated on a 96 well plate and then detected using a labeled reagent. And in this case, we're using the analyte as an antibody. So we can think of any type of assay that uses an antibody or a different analyte. So the same principles are going to be applicable for that kind of ligand binding assays. Slide. So during method development, what's the first thing we do? First, we need to make sure we are very comfortable with the assay context and intended use of the data. So why are we doing this assay? So is it a quantitative measurement of an analyte? Is it a semi-quantitative measurement for pre-existing antibodies in this case? Or is it supporting a selection of patients for clinical studies? Next step, we need to define an assay format. So the assay format can be very variable. So we need to first evaluate what are the different commercial or custom reagents that could be used for the assay. So define which reagent is going to be used as the capture, define which reagent is going to be used as the detection reagent, and what kind of signal are we expecting for the assay, and what's the reporting strategy. Is this a titer assay, is it a measurement, or is it a signal to noise? These all need to be defined very early on before we start the method development. So we're not going into a lot of details into method development because this is fairly standard to optimize the assay, but really the next step is to optimize the assay conditions, capture and detection reagent concentrations, label detection reagent conditions, optimize wash steps, the different buffers, incubation times, and this needs to be done based on results during method development. So it's a series of iteration where you basically run your assay in different conditions and define which are the best conditions. And once you have a good basis for that, you can draft your analytical procedure, which is going to be used for the next steps. So again, during method development, we need to define what are the controls that are going to be used for the assay. So in some cases, we need to identify lots that are negative. So commercial lots, for example, serum samples are negative, and qualify them, and then generate a pool that's going to be used as our negative pool, for instance. We can also use commercial positive antibodies or commercial reagents that can be used, and also use endogenous analytes from these serum or plasma samples. And finally, it could be in-house generated. So depending on your assay format, the intent of your assay, you can use any types of combination of these options to generate the proper control for the assay. And then once you generate these controls, you need to set the control levels so that they represent the expected range of the study samples that are going to be analyzed in the assay. And these can be selected based on the endogenous levels, or they can be spiked to reach adequate levels depending on the assay. Some of these controls can be used as validation controls, and other controls or the same controls can be also used for run acceptance. Again, to be defined based on the assay format and conditions. So these validation controls could be used to evaluate precision and accuracy, linearity, range, stability during method validation. And other controls or similar controls can only be used for controls for run acceptance when you're going to be running the assay for clinical study samples. So once we have optimized the assay, we have our controls ready, we want to make sure that the assay is going to be fit for method validation. So we recommend to run a few experiments that we call pre-validation evaluations, where you're going to be running some validation assessments in the assay. And these are very useful to establish acceptance criteria for the upcoming method validation. And it can also be a sign that the assay will need to be re-optimized and brought back to method development if needed. And once we have all that set, it's time to optimize and clarify your draft analytical procedure, because we want to have the analytical procedure as ready as possible to move to the next step in method validation. Finally, during these steps, it's very good time to train different analysts that will be involved during method validation. So next slide. So during method development, we need to identify what are the critical reagents. We've talked about the capture reagent. Obviously, this is a critical reagent. We've talked about the detection reagent is also a critical reagent. In this case that we are presenting, we're using a commercial antibody or commercial reagent that is custom labeled. So it's labeled by the lab. So we need to identify multiple sources for all the commercial reagents. And you will come back later to that during design control. And then also it's very good to know how many lots of commercial material is available before we move to the next step. So the next parameter to evaluate is identification of the critical reagents, critical steps, sorry. So define which are the different ranges and the variables for these steps. So for instance, incubation times, temperature range that are going to be tolerated and used for the different assay steps. Finally, blood sample collection method. Type of tubes, the procedure, incubation time, and all these things, these are part of critical steps of your assay. So now we're very comfortable with the assay. We've identified the critical reagents, critical steps. We need to prepare a design control plan. This is a documentation to plan the strategy for the design control. We need to set the expected acceptance criteria based on context of use and based on method development results that were generated. We need to set timelines. We need to identify the staff that's going to be involved and prepare that documentation. And that's where a project manager involvement would be also very critical. So next.
Let's continue with the phase 2 of the workflow for the design control. So here, let me summarize what we just saw in plain language. Design control is simply a structured process. It connects what we want to achieve, the benefits of the patients, to what we need to prove, the regulatory evidence. It starts with user needs. Why does this assay exist? What clinical question does it answer? Design input translates that into number, acceptance criteria, threshold, and target for every parameter. This must be defined before any experiment starts, not after. Design process and output is qualification work, reagent, assay parameter, and so on. Verification confirms the priority parameter, it's the predefined criteria. Validation is the full performance evaluation. Risk management under ISO 14971 runs through the whole loop. Every review gate is also a risk checkpoint. And the golden rules here, nothing is tested, nothing is changed without a document rationale. It's not written down, it didn't happen. I will now hand over to Martin.
Thank you, Marie-Laure. So what does it mean, design control in practice? What are we doing in the lab? Next step, please, next slide. So we've talked about blood sample collection step. This is a very important step. So what are we going to do? We're going to take different blood collection tube types and suppliers. We're going to test blood collection in all of these conditions using serum processing methods, for instance. So different centrifugation times are going to be representative of what's expected at the clinical sites. Temperature, centrifugations of the samples, processing time and limits. So these are all things that need to be verified during the design control steps. Then we have a coating capture reagent. So what is the different coating times that the assay can allow? We need to compare different lots of that capture reagents to make sure that all the results will be consistent through different lots. And then we also need to obviously try to get different suppliers and test different suppliers, because if one supplier doesn't have this reagent anymore, we'll need to move to another supplier, and that can be a real challenge. We've talked about this assay as a labeled, custom labeled detection reagent. So the commercial reagent that's used for label, we need to verify lot-to-lot variability. Again, if we can have this reagent from different suppliers, it will need to be tested as well. We need to do custom labeling, so generate multiple lots of the custom labeling and compare these lots in the assay to make sure they are very consistent. Let's say we're using a plate, so we can test different plate lots. We can also use plates from different suppliers. But what happens if there's only one supplier, like MSD for MSD assays? So we need to mitigate these problems as well. So we talked about incubation time, incubation temperature. We're going to need to compare the minimum and maximum limits for all of these different critical steps in the assay. So the assay will also have multiple solutions. So these will be tested through different preparations, again, different suppliers if available, and the in-use stability and the lot-to-lot will typically be confirmed during method validation, because during validation you'll be doing multiple runs and you're going to be using multiple solution lots and multiple conditions for your assay. Next slide, please. So at the end of design control, data analysis, you need to take all that data and look at it very carefully. Set the method limits. What are the incubation times? What are the precision? What are the variability? What is the background signal of different serum samples? What is the variability of your control samples? And then based on that, you can update again your analytical procedure in parallel to make sure that your assay is very well controlled for the analysts that are going to be doing the assay. And then based all of that data, you can set final validation assessment acceptance criteria. So these are the preset acceptance criteria criteria that are going to be used for method validation. So we've talked about this a little bit. So there's mitigation strategy and the justification. So all of these steps may have problems, or expected, problems that you can have. So you need to mitigate what's going to happen for each of these steps. I've talked about, let's say you have only one supplier for one of these reagents. What's going to happen if there's a problem with that supplier? We need a backup plan, and you need to evaluate the risk on the assay and the assay livelihood. So you update your mitigation strategy at this step. You update again the analytical procedure to include lot qualification. So let's say you have tested multiple lots in your design control. So you can see that there's some variability. You might need a procedure specific to qualify a new lot of reagent. So this needs to be incorporated in your analytical procedure. And again, I've talked about this, mitigate the absence of different suppliers in this case. So the definition of design control, it's a structured process linking scientific intent to regulatory evidence. And as you can see, we need to work in close collaboration with the regulatory affairs to make sure that we're all working in the same direction, and we document this in parallel throughout the whole process. So basically, we need to demonstrate with data and documentation that the assay will generate reliable results in all, hopefully, the real-world conditions expected. So once we've done all the design control, we're moving to the method validation. How to preset these validation acceptance criteria. So these should be based on current guidance documents, for instance, ICH M10, CLSI, and these to be applicable to your assay, and again, your context and intent of use of your assay. Your acceptance criteria also need to be based on method development and design control experiment results. So obviously, if your assay is variable in design control, it's not going to get better during method validation unless you go back and optimize your assay. It needs to be based on your assay format again, because not all assay formats will generate reproducible results in the same way. Again, going back to documentation, you need to provide scientific support and justification for all these validation assessments, step, and acceptance criteria. Controls. You need to use control samples for the method validation, and you need to cover the range of the expected results during clinical study analysis. They need to be representative of these samples, and they also need to demonstrate the assay performance on a day-to-day use. Again, these acceptance criteria need to be adapted from guidance document, from method development results, and the context of use of the assay. For instance, the assay needs precision around the threshold where patients will be included or excluded. That's very important, again, going back to the context of use of the assay. Next. So these are examples of method validation assessments that might be needed for different types of assays. So for precision, so the assay needs to be reproducible using the quality controls and validation controls and positive control samples. It needs to be done with multiple analysts, multiple runs, and multiple days. Accuracy for quantitative assays might not be applicable for semi-quantitative assays. Sensitivity is very important, and what's the background signal we're expecting with these assays. So that's very important because you need to stay away. You need to make sure you understand the sensitivity of the assay with the threshold and what's going to be used to establish a yes or no decision. So for matrix selectivity, similarly, we need to do spike and recovery experiments to make sure there's no interference from matrix. And again, more specifically, we'll need to evaluate endogenous, hemolyzed, lipemic, icteric, disease-specific samples that could interfere with the assay format. We can also use, during method validation, we would evaluate exogenous interference from, let's say, concomitant drugs or other analytes that can be present in different disease populations. Establish linearity and range of the assay with the calibration curve, dilution linearity, etc. Sorry. Need to demonstrate specificity, robustness as well. We've also mentioned this earlier, that we need to verify different lots of critical reagents, and all the reagents are going to be useful or can be used in the assay without affecting the results. Different incubation times during PNA, for instance, can be used. We need to evaluate, very importantly, stability. Stability of these control samples can be used freeze-thaw, benchtop, long-term, but also reagent stability. So we're using custom reagents or commercial reagents. We need to make sure that the stability of these reagents is well demonstrated during method validation. So finally, we have an assay, it's validated. We need to validate further, but how is going to be set the threshold for inclusion exclusion? So this is the goal of the assay, is to exclude or include patients and make sure that the patients are used or not in the clinical study. So the clinical utility of the assay will be very important here and will need to be backed up with scientific validity as well. It needs to be based on context of use and set to define the benefits for the patients. The threshold can be specific for different types of clinical study, because for one population, we're expecting lower signal. For the other population, disease population, we can use different threshold. So this also needs to be established very early on. So ideally, your threshold will be based on previous clinical data. You already know from previous clinical data generated with a similar analyte that the assay can tolerate or not that kind of for the patient. So it can be also based on previous literature and much supported by the analytical performance of the assay, sensitivity, especially in precision and reproducibility. And finally, we need to collaborate with clinical sponsor, because they are the ones that know where they want to go with this assay, and we need to set this threshold based on sponsor and the assay and the conditions of the assays as well. So concretely, the threshold is used to exclude or include patients from a clinical study. So the data needs to support that patient will not benefit from the treatment or have high safety risk before being excluded from the assay, from the clinical study. And the assay needs to be highly reliable around that threshold that's going to be set for the assay. So I think the next step, I'm going to back to Marie-Laure.
Okay, thank you, Martin. Now time to speak about the technical dossier. So the technical dossier is the final output of everything we have been building. Let me explain why it matters and why it actually contains other Article 5.5. Why it's crucial? Because it's the document that protects the patients. It ensures the assay gives accurate results, support the right treatment decision, and minimize the risk of misdiagnosis. It's also the document that allows corrective action to happen quickly if something goes wrong after the test is in use. Now, what does it actually cover for an LDT under Article 5.5? The structure follows strictly the IVDR Annexes, 20 sections in total. 12 are formally required. They include, for sure, the GSPR checklist, the performance evaluation, the risk management file, and the post-market surveillance plan. 5 are explicitly not applicable. No damage registration, no UDI, no notified body here, because we are not placing the device on the market. And 3 are not legally required, but strongly recommended as good practice: literature documentation, design change, record, and version control of expired documents. The next slide shows the full table. Here is the full picture. Every section of the technical dossier mapped to the IVDR annexes with its statute under Article 5.5. I want to highlight a few key points. Section 6, 7, 8, and 17 are strictly mandatory. No flexibility there. Section 6 is the GSPR checklist. Every applicable requirement must be addressed. Section 7 is the risk management file under ISO 14971. Section 8 is the full performance evaluation, and 17 is the public declaration, not a declaration of conformity, which is the CE marking document. but a specific Article 5.5 declaration that must be made publicly available. Section 10, 11, 12, 15, and 16 are explicitly not applicable because we not see a marked. Section 18, 19, and 20 are good practice. We recommend including them because they demonstrate a mature quality culture and they protect you if questions arise later. The key message for sponsor here in the room. At Cerba Research, there is how we manage LDT from regulatory strategy to technical dossier, following this structure approach end-to-end. Now back to Lorella for the conclusion.
Thank you, Marie-Laure. And therefore, we would like to conclude with this webinar that there are key considerations, or better yet, lessons learned that Cerba Research has learned over time when embarking into an IVDR LDT route under the in-house exemption Article 5.5. There are several. The first one is, it is key that we embark into an immediate collaboration between interdepartmental teams, and that is bringing in the technical scientific team, the regulatory affairs, quality assurance, the program management or project management, and as well as the sponsor, the clinical team at the sponsor, as well as their experts in the area. Finally, also full clarity on the intended use of the assay and the data that will be generated. Establish at the onset the right foundation of the platform and the assay format that will be used. Because when you start early with that strong foundation, then you can use an iterative process of now enhancing the assay and thereby now being useful to embarking into the IVDR compliance route. If you start with an inappropriate platform and an assay format that really is not suitable, you're going to have to restart all of your development. Whereas with the appropriate platform and appropriate assay format as a strong foundation, then you simply build on this assay and it becomes very much effective, cost-effective, timely effective as well. Understand your critical steps and reagents that will be used in the assay such that now your design control will be very well established. Document in parallel with the regulatory affairs, the technical dossier. Leaving it at the very end will only cause you then to go back and having to restart some of the experiments. And therefore, finally, what is really critical is put the time, the appropriate time invested in your development phase. This is what will set the success for a strong, robust validation and ultimately bring benefit to the patient that really is intended to receive the treatment. And so with this, we end our webinar.